Welcome to the PubMed Daily Review for March 24 ST. Today we scanned 2,310 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5, Adolescent Health, AI, and Surgery. This trend explores adolescent well-being, AI's role in healthcare, and surgical advancements. This cluster covers 510 papers. Representative work includes effects of risk management plus predictive nursing on adolescents with non-suicidal self-injury behavior, the roles and purposes of caring touch in health professional practice, a discourse analysis, and virtual or in-person. A census of orthopedic surgery residency interview formats for Match 2025-inch. Trend 2 of 5, microbial pathogens and antifungal targets. These papers explore cellular mechanisms, host pathogen interactions, and therapeutic strategies for infectious diseases and related conditions. This cluster covers 554 papers. Representative work includes functional and molecular characterization of Aspergillus fumigatus phosphoglycomutase, PGM, a potential target for antifungal therapy, respiratory syncytial virus from discovery to vaccines, and the cytoplasm of living cells can sustain transient and steady intracellular pressure gradients. Trend 3 of 5, neurology, blood pressure, and sensation. This trend explores how neurological processes influence physiological sensations and vice versa, particularly concerning blood pressure and sensory perception. This cluster covers 282 papers. Representative work includes BP metrology, non-invasive continuous blood pressure monitoring to predict hemorrhagic transformation after endovascular thrombectomy, control of innate olfactory valence by segregated cortical amygdala circuits, and alcohol attenuates CRF-induced excitatory effects from the extended amygdala to dorsostriatal cholinergic interneurons. Trend 4 of 5, cancer treatment and surgical innovation. These papers explore advancements and challenges in cancer treatment, encompassing surgical techniques, drug development, patient care, and research collaboration. This cluster covers 306 papers. Representative work includes impact of socioeconomic deprivation in patients undergoing elective surgical resection for colon cancer, feasibility and safety of a robotic-assisted optical navigation system for pulmonary nodule percutaneous cryoablation, a prospective, single-center, single-arm pilot study, and blocking SHB2 benefits FGFR2 inhibitor and overcomes its resistance in FGFR2, amplified gastric cancer. Trend 5 of 5, orthopedics, nutrition, and surgery. This trend explores the intersection of surgical interventions, nutritional impacts, and patient outcomes in various medical contexts. This cluster covers 658 papers. Representative work includes nutritional paradoxes of the new dietary guidelines for Americans, 2025 to 2030, a critical analysis, minimally invasive treatments for benign prostatic hyperplasia, a narrative review, and HOPE trial. Hemiarthroplasty compared with total hip arthroplasty for displaced femoral neck fractures in octogenarians. A 4. And 10-year follow-up of a randomized controlled trial. Up next, our first paper out of 5, protein structure prediction powered by artificial intelligence. From biochemical foundations to practical applications by Tianxiang Yin and Jingrui Liu from Department of Health Technology and Informatics, the Hong Kong Polytechnic University, Hong Kong Special Administrative Region of China. Let's break down its key contributions. Proteins are the workhorses of our cells, and their 3D shape dictates their function. Understanding these structures is key to unlocking biological mysteries and developing new medicines. However, getting these structures experimentally is often slow and expensive. Traditional computer methods have also struggled, especially for new or complex proteins. This is where artificial intelligence has stepped in, revolutionizing how we predict protein structures. This paper is a review, looking back at how we got here and where we're going in protein structure prediction. It examines the fundamental biochemical principles of protein folding and then dives into the cutting-edge AI methods, like AlphaFold and RoseTTFold. These models use deep learning to learn complex relationships between a protein's amino acid sequence and its final 3D shape, integrating vast amounts of data and biological knowledge. The results are truly remarkable. Models like AlphaFold 3 are now achieving near-experimental accuracy, a huge leap forward. They can even predict how proteins interact with DNA and RNA, opening up new avenues for research. While some models like ESM-Fold offer incredible speed, and others like RoseTT-Fold excel at predicting mutations, the overall trend is clear. AI has dramatically advanced our ability to understand protein structures. So what does this mean for us? These AI tools are revolutionizing drug discovery and our understanding of diseases by providing rapid, accurate structural insights. However, it's crucial to remember these are predictions, not definitive experimental results. They serve as powerful starting points for further research and validation. The future promises even more sophisticated AI that can model protein dynamics and guide the design of novel proteins with specific functions. Up next, our second paper out of 5, Quantum Sensing MRI for Non-Invasive Detection of Neuronal Electrical Activity in Human Brains by Yongxian Qian and Fernando Iboda. Let's break down its key contributions. The human brain's electrical activity is key to everything we do, from thinking to moving. But directly measuring this activity in real time, without invasive procedures, has been a major hurdle. Existing tools like fMRI or EEG have their own trade-offs. This research introduces a novel approach to tackle this challenge. This study introduces quantum sensing MRI, or QSMRI, a groundbreaking technique. It uses the body's own water molecules as tiny quantum sensors within a standard clinical MRI scanner. By analyzing subtle changes in the MRI signal, researchers can directly detect the magnetic fields produced by neuronal firing, offering a new way to visualize brain activity. The results are exciting. Researchers successfully demonstrated that QSMRI can indeed detect the magnetic fields generated by neuronal activity directly. This is a significant step, establishing a functional imaging method that doesn't rely on blood oxygen levels like fMRI. The technique showed promise in visualizing activity in various brain regions, and even in a case study involving a neurological disorder. So what does this mean for us? This study presents a powerful new tool for neuroscience and clinical applications. It allows us to directly observe the electrical conversations happening in the brain non-invasively. This could revolutionize how we study brain function, understand neurological diseases, and potentially develop new treatments. Up next, our third paper out of 5, a TGF-beta/IL-2 or immunomodulatory fusion protein transforms immunosuppression into T-cell activation to enhance adoptive T-cell therapy by Yaping Su and Philip D. Greenberg from A Program in Immunology, Translational Science and Therapeutics Division, Fred Hutchinson Cancer Center, Seattle, Washington, 98109. Let's break down its key contributions. Adoptive T-cell therapy holds great promise, but its success in solid tumors has been hampered. The tumor microenvironment is often highly immunosuppressive, largely due to factors like TGF-beta, and it doesn't provide the signals T-cells need to proliferate and survive. We aim to engineer a way to overcome these challenges and boost T-cell effectiveness. To address this, we designed a novel class of immunomodulatory fusion proteins, or IFBs. These engineered proteins are built using components of the TGF-beta receptor and the IL-2 receptor. The core idea was to create a synthetic receptor that could simultaneously block the suppressive effects of TGF-beta and instead trigger the activating and survival signals of IL-2, all within the challenging tumor microenvironment. Our engineered IFBs demonstrated remarkable success. They effectively reprogrammed the suppressive TGF-beta signals into the proliferative and survival signals of IL-2. This led to significantly enhanced T-cell function, improved persistence within the tumor, and ultimately, better killing of tumor cells. These findings strongly suggest a powerful new strategy for improving adoptive T-cell therapy. The key takeaway here is that we can engineer T-cells to fight back against the tumor's defenses. By converting immunosuppressive signals into activating ones, these synthetic receptors offer a novel way to boost adoptive T-cell therapy, particularly for challenging solid tumors. This work lays the groundwork for developing more effective cellular therapies in the future. Up next, our fourth paper out of 5, single-cell-based identification of drug synergy with immunotherapy via tumor microenvironment remodeling by Ziruoi Wang and Yongjun Deng from Basic Medicine Research and Innovation Center for Novel Target and Therapeutic Intervention, Ministry of Education, College of Pharmacy, Chongqing Medical University, Chongqing, China. Let's break down its key contributions. The tumor microenvironment is a complex ecosystem that can hinder cancer treatments, especially immunotherapy. We need better ways to find drugs that can work together with immunotherapy to overcome these challenges. This study aimed to develop a systematic framework to discover such synergistic drugs. Researchers created a valuable resource using single-cell RNA sequencing to study immunomodulatory drugs. They screened nearly 740 compounds across different cancers in mouse models. This allowed them to identify key biological programs associated with changes in the tumor microenvironment and develop a scoring system to measure responsiveness. The study identified 12 distinct metaprograms linked to TME remodeling. A key finding was that allopurinol, a gout medication, significantly boosted anti-PD-1 therapy. This combination resulted in a remarkable 4 out of 6 mice achieving tumor-free outcomes, demonstrating a powerful synergistic effect. This research offers a powerful new way to find drugs that can make immunotherapies work better by understanding how they change the tumor environment. The identification of allopurinol as a synergistic agent is particularly exciting, suggesting potential for repurposing existing drugs. This work opens doors for developing more effective combination cancer therapies. Up next, our 5th paper out of 5. Blood-brain barrier-penetrative lipid nanoparticles enable systemic delivery of TRIM11 mRNA to disaggregate tau in Alzheimer's disease models by Yin Zhou and Bingyan Shi from Hunan Key Laboratory of Brain-Targeted Bionanomedicine, Hunan International Joint Laboratory of Nanobiomedicine, School of Life Sciences, Hunan University, Kaifeng 475004, Hunan, China. Let's break down its key contributions. Alzheimer's disease affects millions worldwide, and unfortunately, we still lack effective treatments that can halt its progression. A key feature of AD is the buildup of toxic tau protein tangles in the brain. While we've identified a protein called TRIM11 that can break down these tangles, getting mRNA therapies across the protective blood-brain barrier has been a major hurdle. This study explores a novel way to overcome that challenge. To tackle the BBB challenge, the researchers developed special lipid nanoparticles called PLMPs. These nanoparticles are designed with a component that helps them cross the blood-brain barrier and deliver their cargo, TRIM11 mRNA, directly to brain cells. They tested this system by giving intravenous injections of PLNP, TRIM11 mRNA, to mice modeling Alzheimer's disease. The key innovation here is how the PLMPs use specific interactions to get across the BBB and into the brain, showing a significant increase in delivery to the hippocampus. The results were quite promising. The PLMP-delivered TRIM11 mRNA showed an impressive 8-fold increase in accumulation within the hippocampus and over 30 times higher neuronal transfection compared to unformulated mRNA. This led to a significant reduction in key markers of Alzheimer's pathology, like phosphorylated tau aggregates. Importantly, the treated mice showed improvements in cognitive tests and even demonstrated a prophylactic benefit when dosed early, suggesting this approach could both treat and prevent disease progression. This study presents a significant advancement in delivering mRNA therapies to the brain. The engineered PLMPs successfully crossed the blood-brain barrier, enabling the delivery of TRIM11 mRNA to combat tau pathology. This approach not only reduced existing tau aggregates, but also showed promise in preventing their formation. While these findings in mouse models are exciting, further studies are crucial to determine if this innovative therapy can be safely and effectively translated to human patients suffering from Alzheimer's and other tauopathies.